Coming to my talk, and I'll be speaking about tissue culture and the role in native plant conservation. You guys, the naughty guys in the back over there. No wonder. Okay. Okay.
There we go.
Okay. Okay.
Yeah.
Okay. I just wanted to give you some history about native plants in Hawaii. There's about 1,400 native plant and about 188 native fern species in Hawaii. And a lot of them exist in small populations and in lower numbers, so which makes them pretty vulnerable to any kind of like disruptions or of habitat or urbanization or even pests that come in. It puts them at risk of extinction, and so a lot of times there has to be some kind of intervention or some conservation work done for some species to save them from extinction. Okay. As far as the Fish and Wildlife Service, they maintain a species list of species that they consider at risk of extinction or decline. And as November 2012, there were about 734 species that they were looking at. About, 270 are federally listed as threatened and endangered. 49 are proposed endangered. In other words, they are pending endangered to be put onto the endangered species list. 18 are candidate. These are in the process of being put onto the federal register. 274 species of concern, and these are the ones that there is not enough information at the moment to actually consider them for listing. But you know what was interesting is that a lot of the extinct species that have occurred, the extinctions, have come from this group, the species of concern. In other words, they had gone extinct before any kind of help they were able to do anything for them. And the 23 are with no status. Okay, on the next slide. Hawaii has its own, consortium for plant conservation. It's the Hawaiian Rare Plant Restoration Group. And it's made out of several state and federal agencies, different public and private conservation groups and environmental groups, private landowners, and local botanical gardens. These entities or agencies, they have one or more roles in fulfilling the mission of the Hawaiian Rare Plant Group, and that's to prevent the extinction of native Hawaiian plants by providing some kind of cooperatively administered recovery or conservation system for plants, doing sampling, propagating, and reintroducing of rare plants, and preserving native plants and their habitats through communication and public education. Okay. My program, the Hawaiian Rare Plant Program, we're a horticultural program, and where we use horticultural techniques to save and do conservation as well as germplasm preservation. Our mission is, again, to prevent the extinction of these native plants and to propagate plants for approved restoration projects and garden use, and also to maintain an in vitro and seed germplasm collection of the critically endangered Hawaiian plants. Okay. Sorry, it's kind of dark, these slides, but again, like the Hawaiian, my project, we use 3 different types of propagation horticultural techniques to save these plants. And one of it is tissue culture, and you can click again. And before I go, in this room alone, there's about 18,000 plants in there, and about 167 species too. So in just a little room that's about 20 by 25 feet, you can put a lot of plants in there. Even though it's pretty high maintenance because it's a controlled environment, you can store a lot of tissue there. You can click the next picture too. We have a greenhouse facility and tissue culture. There's a transition from tissue cultured plants to the greenhouse. It's an important last stage of tissue culture, as well as we use our greenhouses to establish cuttings from the wild as well as seed germination. That is, I think that's Maui, Olinda. Yeah. And the next two pictures. And this is what Alvin will be talking about later, and then the second half is our seed storage program here. So, and that's Alvin looking in the refrigerator. Okay. So I'll be talking about micropropagation, or tissue culture. And we start with different types of propagules to establish in tissue culture. And the sexually derived explants are usually mature seed. We can extract embryos and ovules from the seeds if they're like immature, or maybe they're not just the perfect or the optimum health. The seeds are kind of like compromised. We do fern spores, and we can even germinate pollen to get plants. Okay, next slide. We can do clonal propagation using vegetative plant tissue. We commonly use apical meristems, which is the growing tip of a plant, axillary meristems, which are the lateral buds, which fall on the side here, root meristems, stem internodes, which they have no meristematic tissue, they're in between the nodes, and inflorescences, as well as leaves. We can use them for clonal propagation. Tissue culture, or having successful tissue culture, begins in the field or in the greenhouse. To have like suitable plant material is really important. You know, some plants respond to different types of tissue culture processes or techniques better using a certain way, and some are better another way. So sometimes, like maybe using seed embryo to germinate embryos rather than ovules are better, or doing clonal work, or you know, it's just choosing the right material for the right technique to get the plant growing. Post-harvest handling is really important. A lot of people, they do the perfect collections and they neglect this part, and they compromise the material by storing it wrong before they bring it into the lab. Proper surface disinfestation. We have to sterilize the material before they come into the lab, and in that process we can actually kill the tissue. And plant medium and culture conditions. This is all aseptic when the plants are within the tissue culture lab and they themselves are growing. It's just like in the greenhouse, they're growing medium and putting it under the right light and temperature regimes. So to go more in depth, you can go. Selection of suitable plant material. The time of harvest is really important. You don't want to choose cuttings or seed collections at the wrong time of the year. A lot of times people will bring in cuttings that are from things that have terminally, they're ended, the vegetative state has ended, and they're in the flowering stage, and they're really not suitable to get vegetative tissue culture going. Juvenility, it continues on into the plant tissue. If you pick material that comes from the mature part of plants, which tends to be on the upper part of the trees or the inner branches, it's really hard to start these. So like a lot of times when you're doing cuttings, and it might be even, it pertains to greenhouse cuttings too, you want the juvenile parts, which are the ends of the trees, or on root, like shoots, or on the stump, or on stumps. So these lateral shoots that come out are better. The general health, the healthier your plant tissue, the easier it is to get tissue culture to be successful. Post-harvest handling. You don't know how many times I've gotten plant material come in from collectors that went hiking for 5 miles, put them in a plastic bag, put it in their backpack, stuck it in the sun, went looking for other stuff, come back, bring it back to the car, and then bring it in 2 days later into the lab, and I get this mush bag of like cuttings or fruit. And to do that much investment to do collections of material, and sometimes a really rare or really important plants, you know, it just makes sense to just pay a little more attention and a little more care for your plants. So bring a pack of an ice cooler or something, and just get it into the lab as soon as you can, or even in the greenhouse. And that will help a lot with excessive temperatures, anaerobic conditions, microbial growth, and damage. Before it can even go into the tissue culture, test tube, into the sterile condition, we have to do a pre-treatment. And that's just any plant tissue that you bring from the wild or you bring in from the greenhouse, they have a microflora on top. It's just like our skin. We have microflora just growing on us. So we do like these pre-treatments, like water rinses, just simple water rinses. We have an ultrasonic cleaner that you clean your jewelry with, and it kind of breaks the air bubbles. It's especially useful when you have plants that are hairy, and you get the bubbles out between the hairs. And ethanol dips, 5-sand soaks, and zerotols are really good. And this is not sterilizing the tissue, it's just knocking back that microflora on the tissue. Okay. The actual sterilization treatments are pretty harsh on the tissue. So there's a fine line from getting the plant tissue sterile for tissue culture to make it aseptic and to killing the tissue. So, but the common cleanser we use is just a plain Clorox bleach at a 5 to 10% soak. Plant preservative mixture is a biocide that you can buy. It's really expensive. It's like $1 a mil, and you usually end up, it's really expensive, but you usually end up using about a 5% solution, so it can be costly to do this. And chlorine gas sterilization, which is the picture in the back where we're actually making a fumigation chamber and putting the seeds on filter paper like this, and then inside here, this liquid is a 85% Clorox bleach solution, and you drop in 1 drop per 100 mils of Clorox solution of hydrochloric acid. Then you cover it and seal it and put it in a dark. And what happens is it causes an explosion of chlorine atoms or ions to be released, and it's like a fumigation. And this has been really successful. A lot of the seeds that we were not able to get clean, because if you look at seeds under a microscope, they have all these invaginations, they have little sometimes sheets covering like that. These small little molecules of chlorine are able to get into it and sterilize it. So we fool around with plant media a lot. A lot of the native plants have no propagation protocols at all. So we start off usually with 4 or 5 different type of formulations. And Murashige and Skug is the most common plant media ever. For any plant around the world, they usually start with Murashige and Skug, woody plant medium, Knudsen, and we modify our mixes quite a lot. So those are the planting medias that we plant in tissue culture. One technique we use a lot for hard seeded coats that have just a physical dormancy, and this is of Carnavalia, it's a Fabaceae. We actually take that seed and we'll dip it into alcohol and flame it. And what happens is it cracks, makes little micro cracks on the seed, and it breaks that dormancy. And so we get like germination within like 3 days, and a seedling like this. So, and then on top of that, it sterilizes the seed so we can put it into a sterile environment. Ovule and embryo culture. I don't know if people who grow Pichardias, you folks know that, a lot of them, they take a long time to germinate. But I started this process because a lot of the wild Pichardia were getting predated, eaten by like rats in the wild, so they never could get mature fruit. So they'd send me in these immature fruits, and I tried to propagate it by just taking off the husk and planting this, what is that, embryo tissue, and the endosperm and the little embryo, but it would rot because this part would rot. So I started to excise the embryo here and plant it into a test tube like this, and that's embryo culture. I can get a Procardia to germinate like in 1 week doing this. So it's really efficient, and it's really pretty simple, but they don't like staying in a tissue culture tube like this because they grow pretty fast. So usually within like about 3 months, I have to take them out and put them in a greenhouse. But you can make germination of Procardias pretty rapid by doing this technique. Organogenesis is a clonal propagation technique where you're cloning actual pieces of cuttings. And again, we use a modified Mirshigan skug usually, and we kind of manipulate it with auxins or cytokinins. And we do what is called direct organogenesis, or shoot organogenesis, where we're trying to avoid any kind of callus stage because we're trying to avoid any type of mutation or excessive growth. And what is inside this test tube is a Hawaiian mint. And it's really, really easy to propagate by cuttings. And the reason why, and it's really easy to grow in a greenhouse too, but this is an Army plant that is monitored by the Army. But they put it back into tissue culture, and we maintain cultures in germplasm storage like this because they get hit by white flies and mealybugs. They're really susceptible to all the different types of introduced pests, so it's safer for them to stay in tissue culture. We do a lot of fern spore germinations, and Hawaii and our ferns, we have 2 different types of ferns. Ones that produce the green spores, this yellow type, and it actually has some chlorophyll in it, and they cannot store well. These are the black spores, and they can store for a long time. So I just wanted to show you these 2, and so we do both types. And you can show in the next picture. So as you see, pala'a is actually, this is all the germinated gametophytes that have occurred, and they're actually a black spore type. And then we can get the next picture. We can get spore, the actual ferns or the sporophytes come about pretty easy and propagate this way too. The last stage of tissue culture is the greenhouse establishment. It doesn't end until we can successfully get it into the greenhouse. And so we use our greenhouse to establish not only the tissue culture, but to grow cuttings that like are excess when we work on our tissue. And this is the end product that we hope to and desire, is to have them put back into the restoration or back into the wild. It's not complete until we have that full cycle. I just wanted to show you some examples of how we used tissue culture to get some, to work with some of our endangered plants. This is mehamehame. It's a Flugia neovavra, and it grows on all the islands except, I think, Lanai, and it's extinct on Molokai. It's really endangered. There's only about 15 populations of a total of about 100 plants left. It's dioecious, so it has male and female plants. It's heavily, heavily affected by twig borers, and so most of the plants are relic plants that are declining severely in the wild. So this is how most of them are in this kind of state right now. So we can grow seeds in the green in tissue culture, which is pretty easy. And you can keep on clicking. And we eventually plant them out to flats like this, and then they can be grown up and put into restoration projects. And also we started to use grafting techniques on Oahu. The Oahu, there's more Oahu plants, in other words, and seedlings. And whereas the Maui plants, there's only about 2 left in the wild. And so what we're doing is we're taking cuttings from the Maui ones and grafting it onto Oahu rootstock, just trying to increase the numbers of the Maui population. Kokea kukiai, or kokio, is actually extinct in the wild, and the only reason why it still exists in cultivation is because of cuttings being collected off of a tree that was growing in a path of a fire. and they grafted these cuttings onto a sister species, which is Coquilla drinarioides. And Waimea Arboretum had been maintaining this plant stock for a number of years. They never produced, you can go on, like viable seed ever. And you can click. And you can click again. Keep on clicking. You can click them all. Okay. This is the kind of seed that they would produce before they ever matured, but every once in a while they'll throw something that would come to near maturity. So what we started to do was excise the embryos and do embryo culture again. And this is a Malvaceae, by the way. I get them to grow in tissue culture, and then we have plants that have been planted out. There are at least 6 seedlings out now there, and a couple are reproducing and producing seedlings of their own now. So, you know, it's almost becoming full cycle for this plant. Okay. And this is my last slide. I just wanted to show you some of these really, really rare plants that had gone through the tissue culture process. Cyanea superba. This is extinct in the wild. They're reproducing populations now on Army restoration sites. This is Cyanea grimsiana, used to be found in the Kola Mountains. It's extinct in the wild. And they just started a restoration project in Manoa Falls Trail. Oh, that's Pinnatephida, I'm sorry. That's extinct in the wild. This is superba. This is Brighamia insignis, found on Maui, Kauai. There's only one plant left. And this is Cyanea truncata, where it was extinct in 1970, refound in the 2000s, I think, went extinct and then found again. So there's 2 plants known in the wild and a bunch of seedlings that they started to do restoration work on. So, and that's about it. So I'll turn it over to Alvin.